Okay, so let's recall the events of the resting membrane potential. Okay. And now we know that when we speak about this cell, so we'll talk about a cell here. And the cell in question that we're speaking about is a neuron. Okay, so let's just bear that in mind. We'll talk about the structure of a neuron here in a minute, but we know that a neuron is the resting cell that we're speaking about. We also know that the inside of the cell is more negative than the outside is, right? The outside is more positive. And so this is at rest at negative 70 millivolts. Okay. And so what we'll really review here is how did we get this negative 70 millivolts? How did it come about? Now we know the two ions that we're taking into consideration are sodium and potassium, correct? Sodium and potassium. So let's just make a little key here. This will be potassium, and then this will be sodium. Okay, and so we know that the relative concentrations of sodium and potassium are such that there's more potassium inside the cell, so let's just fill this in. And there's more sodium outside the cell here. Okay. We also know to be true that there are some different permeabilities for these ions. So the membrane is more permeable to which of these ions? To potassium, that's correct. So the membrane permeability, and you'll see me indicate that with this large P, the membrane permeability for potassium exceeds the membrane permeability for sodium. Okay, and we know that to be about 25 times more permeable to potassium than sodium, right? Meaning that there are more channels, or as I compare that to more doors or more doorways. Okay, now the second thing we'll take into consideration is the varying concentrations of these ions. So we're going to look at the chemical driving force here. Right? But before we do that, let's draw some of the structures that we know to be on this membrane. So what's the first structure responsible for the relative concentrations of potassium and sodium across the membrane? The protein potassium pump, right? The sodium potassium pump. So let's draw that here in the membrane. And we know that that is pumping sodium and potassium, right? It's going to pump sodium out. We got 3 sodiums going out, and we have 2 potassiums going in. Right, that's our sodium potassium pump. We also have some leak channels on this membrane, right? So we have, again, representing those varying permeabilities, we'll put 1 sodium channel here. And we know that we have many more potassium channels. So we'll draw a few of those on. Right, and this is reflecting the increased permeability of the membrane for potassium. So in considering our CDF, the chemical driving force, we know that ions are going to move down their concentration gradient, right, from a place of high concentration to low concentration. And so the movement for sodium will be in what direction? Into the cell, that's correct. So there's going to be a chemical driving force moving sodium into the cell here, right, CDF, sodium. Whereas for potassium, there will be a chemical driving force in what direction? Out of the cell, right? So higher concentration inside, so potassium is going to want to move out. Okay, so let's just make a note of that here. Sodium has a direction into the cell, and potassium has a direction moving, you see that, moving out of the cell. Right, that's our chemical driving force based purely on our concentration gradients. Let's look at the electrical driving force here. So that's sort of step 3. Okay, now what determines the electrical driving force? This we talked about in Unit 1. What determines the electrical driving force? What determines the electrical driving force? Correct, so that difference in charge between the inside and the outside of the cell. And we said that the sodium-potassium pump is what's really responsible for this difference. Okay, it's pumping 3 sodiums out, 2 potassiums in, and there's that excess positive charge leaving the cell, right? And so we develop this environment inside the cell that's more negative. And we know that our electrical driving force is based upon this polarity of the cell, that inside's negative and outside is positive. Okay, so looking at our electrical driving force, we must take this polarity into consideration. And so what are the directions we're going to have our ions moving along their electrical force now? What about sodium? What direction will sodium move in? Along its electrical, still inside, that's correct. So let's just make another arrow here. Sodium still wants to go inside along its electrical driving force. Okay, so sodium into the cell as well. Potassium, on the other hand, is going to do what? It's a positively charged ion. The inside of the cell is more negative. Potassium will likely want to also go into the cell, right? So because these are 2 positively charged ions, they're going to actually be trying to get into the same place. They're going to be trying to get to this opposite environment inside the cell, which is more negative. And so we have an electrical driving force for potassium into the cell. Okay? So let's make a note here, potassium into the cell. Okay, so we looked at the chemical driving force and the electrical driving force separately, and we know from our prior knowledge that we have to combine these forces and look at the E, C, D, F, right? The sum total of these 2 forces. And when we do that, we know that if the direction of the 2 forces are the same, we simply combine the magnitude of those forces. Okay, and by that principle, what is the movement of sodium going to look like in terms of its magnitude and then in terms of its direction? If we're combining these 2 forces. So if we're combining the forces, right, it's simply going to be a larger force. So we're going to have a stronger force, right, in terms of the magnitude or the strength of that, that movement, right? So sodium has a stronger force, and the general direction will be into the cell. Okay? So sodium is sort of straightforward in this way. We have the direction of both of these forces in the same direction, and so we simply add them, and that becomes a bigger force. When we looked at the numbers on our slides last week, that ended up being 130 millivolts of a difference. Okay, combining that 60 with 70, like the resting. Okay, with potassium, it's different because we have forces in opposite directions. So what's going to happen with these two, combining these 2 forces? What's going to happen with combining these 2 forces? Very simple, right? We'll subtract the weaker force from the stronger force. So that will tell you what the magnitude will look like. It will not be as strong as sodium's ECDF, right? Simply because we have forces going in opposite directions. So we'll subtract the stronger force from the weaker force from the stronger force, which will give us a smaller magnitude, and then the overall direction will be in the direction of the stronger of those 2 forces, okay? And so from that, we know that the CDF is going to be stronger. There's a stronger concentration gradient here. And so the general direction of potassium will be out of the cell, but very weakly, not as strong as the force that's bringing sodium in. Okay, so for potassium, we have a weak force out of the cell. I hope everyone can understand how these two forces differ. Yeah. Because we look at the equilibrium potential for potassium, and so the difference in concentration is stronger than the membrane potential at that time. Remember, we said that if we have two different forces, we then look at equilibrium potential versus membrane potentials. Okay, I hope that made sense. Any other questions at this point? Any other questions? Okay, so here we approach what is step 5, so to speak, and this is where the neuron gets to steady state, and that's your negative 70 millivolts, right? When the equalization of these ions happen, meaning that there's no net accumulation of these ions, right? They approach this steady, it's not equilibrium because we're taking more than one ion into account. We're looking at both sodium and potassium. So we won't describe that as equilibrium. Instead, we describe that as steady state, okay? And that basically means that the ions equal out their movement so that that membrane potential stays the same. That's how we get that negative 70 to sort of stick and stay there, okay? That's how we establish the resting membrane potential. Any questions on this? Any questions on this? Again, this is very important for you to be able to draw out, right? You'll get questions on this, right? What is the concentration gradient of the ions? And just having this as a reference point is going to be helpful on your test, okay? Let's go on. Before we get into action potentials, I think it's important that we review the structure of a neuron. Let's talk about some varying parts that I'll mention today. As well as just introduce the concept of different types of potential. So let's talk about potentials here. Okay. And so again, we're speaking specifically about neurons here. So let's look at the structure of a neuron. Okay. And this should really be a review, right? We should all be familiar with the different parts of the neuron. So let's move through this quickly. So we have basically, this is our neuron in question. We have some other axon terminals synapsing onto this neuron. And let's talk about the parts of this neuron in question. So this general region here is called the soma or the cell body. This is the region that receives inputting signals and stimuli from other neurons. And then attached to that are the dendrites. Okay, also responsible for picking up those signals. Then we have the length of the neuron, which is called the axon. Right, this entire length is called the axon. This is where we have electrical signals being propagated to get to the end of the neuron. And the end of the neuron is called the terminal bouton, also called the axon terminal. Okay, axon terminal, also called the terminal bouton. Right? And so the direction of movement of these electrical signals down the neuron takes place from the proximal end, which will be from the soma or the cell body, down towards the distal end, which is the axon terminal. Okay. Now there's a very important region right at the neck, so to speak, of the neuron. This is called the axon hillock. The axon hillock. And this region is very important. It's like a gatekeeper for this neuron. It's going to be responsible for summing the various potentials. So we have electrical stimuli being received on the soma or the cell body. They're going to be summed or sort of analyzed at the axon hillock. We'll determine at this point whether or not we have sufficient change of the membrane potential to actually fire an action potential. So this is going to be a gatekeeper of sorts of whether or not an action potential is propagated down the length of the neuron. okay? And so that's the axon hillock. So we'll speak about that a little bit today. Also, we want to introduce the concept of this region. So think about two neurons that are communicating. We call the first neuron in that chain of communications the presynaptic neuron. The presynaptic neuron, right? And then we call the neuron that's at the distal end, or the second neuron in this chain, the postsynaptic neuron. Okay? And so as a result, we can describe this area where both of these neurons communicate as the synapse. So this area of communication is called the synapse. This is getting a little messy, so I apologize. Okay? So we have neuron 1 speaking or communicating to neuron 2. Neuron 1 is called the presynaptic neuron, meaning before that synapse. Neuron 2 is the postsynaptic neuron, distal to that synapse. And so we have signals moving in this general direction. All right, any questions on that? Any questions on that bit? Let's speak about the different types of potentials here. Let's sort of isolate where along the neuron they take place. So again, looking here at the axon hillock as this area of demarcation, before the axon hillock, we have graded potentials. Okay, and we'll speak about what classifies a potential as being graded. And then beyond the axon hillock, we have action potentials. Okay, action potentials. And so again, this axon hillock is a very important area of deciphering whether or not we have substantial change in the membrane potential to actually propagate or to fire an action potential, right?